Hi everyone, welcome back to another video. So today I wanted to do something different. I wanted to go full science mode on, but again keeping it simple so that you can follow. And again, I hope that this can be a pretty nice challenge for you to fact check me, to do your own research, to try to see if there is actual evidence of what I'm gonna argue that there is no evidence of. And again, why this macroevolutionary theory that attempts to explain how life evolved on Earth and how all living organisms, the rich biodiversity that we observe today, came from a single cell, non-complex, simple organism. And then over billions of years of small changes to DNA, adaptation, natural selection, random mutations, these changes added up and then created these new species, new features, new functions, and again, these different living organisms, more complex, that then became more and more and more complex, eventually leading to reptiles, birds, mammals, and human beings. So is this image here true? Is this what the evidence suggests? So is this a reasonable conclusion from the data and from the evidence that we have today? And again, this is 2025. And again, I'm going to argue that this is not the case. Do not, again, believe me just because I'm saying so. But again, do your own research. Fact check me. Tell me if I'm saying something wrong. I'm mischaracterizing the evolutionary process, or I'm doing some mistakes. I will be more than happy to correct myself, to explain myself a bit better. You see, when doing these videos, you cannot really remember everything or to say everything. So again, just bear in mind that. So let's take a look at some definitions, because again, evolution, the concept of evolution is so wide that we have to dive down. And again, evolution in terms of biology refers to a change in the genetic makeup, so genetic material, of a population over time. But again, it's very important that there are two different types of evolution based on the scale in which they happen. Macroevolution refers to large-scale changes that occur over extended time periods, such as the formation of new species and groups. And that is what I'm criticizing. That is what I believe has huge flaws and has huge problems in light of all the genetic research that we have today, not all we know about DNA, about how changes to DNA work, and so forth. This is this, right? And instead, the other scale of evolution is called microevolution. And this instead refers to small-scale changes that affect just one or few genes and happen in populations over shorter timescales. Microevolution is what we can observe in labs, what we can recreate in labs, or we can observe in actual living population in organisms. So we see these mutations, changes on the DNA, genetical recombination, genetic drift, speciation. All these things we can actually observe. Epigenetics, variation in the expression of certain genes, homozygotes, heterozygotes, all these things, we can actually observe them, we can measure them, we can actually quantify them, and we can see how much it actually affects. And this is actually the basis of what's all the study about genetic diseases, for example, how we can prevent these diseases. Can we do anything about that? Can we change these mutations or these issues that happen in these DNA parts? Can we do anything about it? And again, this is so important. And again, this is well established. But how are they linked between the two? Well, evolutionary scientists actually affirm this. Microevolution and macroevolution aren't really two different processes. They're the same process, evolution, occurring on different timescales. And now here is the very big claim. Microevolutionary processes occurring over thousands or millions of years can add up to large-scale changes that define new species or groups. Is this true? So from the evidence, from the data, from what we know about microevolutionary processes, because again, we have studied this and we still are studying, and we're discovering tons of very fascinating stuff. So we know this. So can these processes occurring over a very long time period, can they add up? See, this is a very weird choosing of words, right? Add up. So these mutations, these changes to DNA, adding up and somehow able to create these large-scale changes, these alterations to the structure, to the behavior, to the metabolic, to whatever is what we're analyzing of the organism, right? That therefore makes it then a new species, that makes it to turn into something different, right? To evolve in this macroevolutionary process into something more and more and more complex, eventually leading to all the rich, complex biodiversity that we observe today. So is this true? Is there any evidence of this? And again, I'm going to try to go through some of the evidence that many times is given that supports this, right? And again, I'm going to try to show you why instead this is not the case. That the evidence given for macroevolution, right, is actually evidence of microevolution. Now, a little bit of biology. I'm not going to go into much detail, but again, this is super fascinating. This is actually how our body stores pretty much the genetic information, right? Then when it's time to either open up, to code, to transcribe certain parts of DNA because there is a certain information, something needed, right? This is the DNA helix, and eventually we get to the genetic coding. Now it's called code because, very interestingly, these nucleotides, which look like this, right? These are chemicals. They actually they are paired always one with the other, right? And now the order in which these nucleotides are paired is super important because that's actually the information that then is transcribed into proteins, into regulation, into different expression of certain gene, and so forth. So the order of these letters is not random. It's important. And the more we discover, like there are tons of things about DNA that we still don't know, but it's very cool that what we do know about DNA is that these letters are not in random order. They're in an actual order that has a purpose. And we might not know all the DNA, why it's coded, and if it's actually important, but we know that it's important. And we have actually classified many places of the DNA, different chromosomes, right? We have 23 different chromosomes, human beings. And so depending on different type of chromosome, there might be some information that we know that particular part of the DNA codes for a certain gene or certain protein, then it's important because it's involved in maybe some genetic disease or something that then we have studied. And therefore, it's very important. And it's very interesting that also these nucleotides are always opposed in the same way. So the adenine, the A, is always opposed to the T, the thymine. And the guanine, the G, is always opposed to the C. So they are paired between them. And when, again, some problems happen on the DNA, sometimes there might be mismatches. And so there might be some nucleotides that are paired with the wrong nucleotide. But again, that's a mistake. That shouldn't happen. And yeah, it's very cool. So that's the DNA. The DNA is just an acronym for deoxyribonucleic acid. Now, instead, the RNA is a single strand. And again, there are different types of RNA. And RNA is more of the active part. Like the RNA is coded from the DNA and then is used to translate it into the actual peptides, so the proteins, right? And on the RNA, we do not have thymine. We have instead the uracil, which, if you just compare the two, is very similar, right? It just doesn't have this group right here in the top left. So the uracil is gonna be paired with the adenine, as it's transcribing for the protein. And this is how the protein transcription looks like, right? Transcription of the RNA, and then the translation of the RNA, the codons. So that's why the triplets, that's why the order of the letters is important, because each codon is going to translate into a particular anticodon, which has amino acids, and then these amino acids are going to put linked together, depending on the order, in the ribosomes, and then it's going to be the protein that's going to be created, right? Again, it's a super summary, but this just to show you the complexity and how fascinating this is. And again, there are tons of different things here that have to work properly. There has to be the protein that opens up the DNA. There has to be the protein that transcribes RNA, messenger RNA into the specific part of DNA that's important, that it needs to be transcribed in order to be translated into a particular protein. The anticodons here, and everything of the ribosome that has to work. And again, tons of different proteins involved, regulation involved in this process. And again, super, super cool and fascinating. Mutations, right? This is what mistakes happen. So the DNA, as it's duplicating, there is also other things like recombination, damaging of the DNA because of radiation or because of the oxidative processes that happen normally in a cell. All these things can damage DNA. But again, mutations is the most common random process that occurs. And again, there are different ways in which mutation can happen, right? The triplet, in this case, that is eliminated, or it's something inserted, or there is a substitution. You can see there's three different things that can happen. And so the DNA might be changed. Now, there is DNA, because of these mutations or because of these errors, these mistakes, or this damage can happen often. We have a way in which we repair it. So there are certain proteins that when they see that something is not working, something is wrong, they intervene and they try to repair the damage that they can see. Of course, this is not perfect, and so sometimes these mutations become fixed. So that means that they're not anymore corrected, and these mutations can be then transcribed more and more and more, and eventually, if they are in a place of specific importance, it can lead to certain changes, certain malfunctions, or as evolutionary scientists say, they can also lead to acquisition of functions. So again, I'm going to try to go through the certain definitions here. Again, mutation, how it's explained, what we see from the data, and then try to take a look at what certain articles say, certain evidence says, and again, if the evidence actually suggests or it can lead you to believe that this process actually happened. And now let's take a look here. Mutations. So it's an alteration to the sequence of the genome of an organism, right? And again, it's can result from errors to the DNA, right? Mitosis, meiosis, and other types of damage to DNA. Now, very important to remember that mutations that are important for evolution, right, are only the mutations that can be transferred into the offspring. So a mutation that happens on the DNA of an organism that is not then transmitted to the offspring is useless in terms of for evolution, right? For this macro evolution to happen, even for micro evolution. So it has to be something that then it can be transmitted to the offspring. And very interestingly, it says here the mutations may or may not produce detectable changes in the observable characteristics of an organism. So play a part in both normal and abnormal biological processes, right? Evolution, cancer, development of the immune system, and so forth. And again, here it says mutation is the ultimate source of all genetic variation, providing the raw material on which evolutionary forces such as natural selection can act. So that's why I said before, this is the main. There are other things in which I have it right here, right? Yeah, in which genetic variation can happen, right? Genetic recombination. There's also genetic drift as well. So there are different ways in which the genetic variation can happen, but in terms of evolution process, in terms of creation of new genetic material, mutations are the foundation, right? And by foundational, they are also random. So these mutations are random, but then they are acted on natural selection in terms of the environment. So specific scenarios may favor certain variations because of their adaptability, because maybe those organisms can survive better. So by surviving, then they can give their genetic material to their offspring, and therefore they can keep that particular feature, right, the variation that helps them to survive. So that's how natural selection acts. And again, it says mutation can result in many different types of change in sequences. Mutations in genes can have no effect, alter the product of a gene, or prevent the gene from functioning properly or completely. So those are the only things that can happen. Mutations can occur also in non-genic regions. And look here, this is very interesting because again, it says 2000 study on genetic variations between different species of Drosophila suggested that if a mutation changes a protein produced by gene, the result is likely to be harmful. with an estimated 70% of amino acid polymorphism that have damaging effect. So what happens is that because these proteins here, these triplets are damaged, right? They're mutated. The proteins then that are created are also alterated. And this alteration can cause, as I said before, the protein to have no effect. So the protein is still working as before. Maybe it's working too much, or maybe it's not working at all, or it has a different, like it's kind of does its function, but just half, or not enough. Or maybe it starts to be bound to something that shouldn't be bound. And again, all these things says here that the result is likely to be harmful. So this polymorphism, due to the mutations, right, that happened, most likely harmful. You can see here almost 70%. So that's a lot. Again, damaging effects. The remainder, so the remaining 30%, being neither neutral, so again having no effect, or marginally beneficial. So of all these mutations that were studied in this particular study, most of them were harmful. So again, in terms of evolution and natural selection, would have been negative. So those particular organisms would have just died out. If either they were neutral, so they had no actual effect, or marginally beneficial. And again, so only very few percentage of those mutations were actually beneficial. And again, that's the Drosophila, right? That actually reproduces in a certain way. More complex organisms that reproduce differently might not have this ability, because again, mutations, if they do not happen in the germinal cells, so the ones that then are going to be transmitting the genetic material to the next offspring, to the next set of population, well, in that case, any other mutation that doesn't happen there is kind of lost, right? It's not fixed, and so again, that can lead to something that then, evolutionary speaking, is not really useful. So yeah, then I wanted to show you here again, always in mutation, right? Again here, by introducing novel genetic qualities to a population of organisms, right, because of mutations, then novel mutations play a critical role in combined forces of evolutionary change. And however, the weight of genetic diversity generated by mutational change is often considered a generally weak evolutionary source. And again, here it says that factors such as natural selection, genetic flow, and genetic drift also are very, very important in the genetic variation, right, that we observe. And now, very interestingly, here, like, mutations are used as if, even though the vast majority of times they are harmful, they have damaging effects. And those very small cases in which there is a marginally beneficial effect is used still to say that they play a critical role in these changes over time, in this macroevolutionary process that eventually can lead to new species, new features, new structures in the organisms, right? And again, here they are very studied because, again, mutations in certain parts of the DNA can cause genetic diseases. And again, there's a lot research being involved here because, again, it's a very good way in which we can prevent maybe diseases. We can maybe change some things. And again, learning more about DNA and about these mutations and how many times these mutations can actually be transmitted. And so that's why there is familiarity for certain genetic diseases. Thankfully, also, we are diploid, right? That means that we have two alleles of the same chromosome, in a sense. So if we are, do you remember Mendel, right? The dominant and the recessive character. So the dominant, if there is a dominant and a recessive, the dominant is the one expressed. So the dominant gene is the one expressed. If both are dominant, that's fine. If one is dominant, one is recessive, the dominant is expressed. If both are recessive, that's when these the kind of less common character comes out, the phenotype comes out. And now, if the mutation or if the problem happens in a recessive allele, that means that the homozygotes dominant or the heterozygotes are never going to express the disease, but the heterozygotes are carriers. So if they have sons with other heterozygotes, it could happen that one of the children is going to inherit both alleles that have the disease, and therefore that they could manifest the disease because it's a recessive disease. Some other diseases are linked to dominant alleles, and again, that's the dominant genetic diseases that are much more common or much more present, And again, that's how we know about these things. And another thing that I wanted to say here is that the Darwin finches. Now, if I go back to I don't know if this no wait, it's yeah, it's here. Like these are different examples sometimes given for mutations, right? And again, these are given many times as example of macroevolution. Even the Darwin finches here, this sometimes is used as macroevolution. So saying, see how the birds evolved, and depending on the food source, they had different beaks, right? They evolved these beaks. Well, did you know that once we started studying the molecular basis of this beak evolution, we actually found that there is a bone morphologic protein, right? That a gene that is found there that has differential expression during development. That means that as the bird is developing, it has different expressions. Sometimes the gene is expressed a lot, is expressed less, is expressed in the middle, and this results in what? Variation of beak size and shape among finches. So this particular gene acts in the developing embryo to lay down skeletal features, including making the beak stronger. And again, it says that also these different beak shapes are also influenced by slightly different timing and special expression of another gene called camodulin. So what happened is that these birds, they had different, where do I have it here? No, I don't remember. Yeah, I think I have it right here. Yeah, this one right here. So the gene is already there, right, for the beaks. And there is already variation within the species. There is variation in how the size is already, because these two genes are there. And depending on how they are transcribed, how much they are expressed, oops, yeah, I don't know why my mouse does something like that, but how much they're expressed actually makes the beaks to already be different. Now, what happens is that the environment, right, forces a certain population with certain beaks to be able to survive, right? Because if the food source changes, then only the birds that have certain beaks might be able to feed and therefore to survive. Those who have like larger beaks might not be able to feed, therefore they're gonna die out. Those who have smaller beaks might be able to feed, so they might be able to survive. So we see the population of finches becoming primarily small beaks because again they can survive. But it's not like the birds are evolving, so their beaks are changing, so in that way they can eat, they can get their food source. The variation for the beak is already there. The gene is already there. Everything is already there. What happens is that the natural selection of the environment is acting on the different shapes of birds, and only those with the smaller beaks can survive. Maybe in other situations, only the ones with the larger beaks can survive. And so we see maybe this drift depending on the environment, right? We maybe see this drift between smaller beaks and larger beaks, but it's not like the birds are changing their beaks. The different size is already there. Depending on the environment, it might act on one or the other shape. So again, a small beak bird may produce an offspring that has large beaks. So it's not like the small beak birds are only going to produce small beak birds. Exactly as a human being that it's short is not always going to produce a human being that is short, right? Could be their sons are taller because maybe in their genes there might be a gene that is expressed differently, so their children might be taller. Again, that's just an example that this is microevolution, okay? And this is not macroevolution. So many times, if we go again here, okay, and it's very interesting because again, this article here like suggests these finches of like these finches, this different big size as example of microevolution. But then if you keep reading, if you go down here, it says Galapagos finches mentioned above. Here are 4 additional macroevolution examples. But again, this is our microevolution, right? It's not macro. The finches did not become another species or not finches anymore, something different. They still remain finches. So, and again, we have to be careful in defining species because this is a term that we have as humans we use to classify animals, but like in the natural realm, there is a continuum. So like animals are not so split. Like a very common definition of a species is that if they produce fertile offspring, then that means that they're the same species. But for like extinct species, we cannot really use this. So again, other definitions come in with the ones with, like, for example, DNA sequence, karyotype. So these are the chromosomes, right? Morphology, behavior, ecological position, environment, and chronospecies, right? And all these things come in place. And again, we might define the different species. Something I said is actually the same species, biologically speaking. So again, remember always that many things as we observe or we define, we cannot really know exactly how they're linked or the exact relationship between them. Right? Because again, we find these fossils, but we don't know how they're related. Which one came first, which one came after, which features appeared before, and which features were inherited or were passed on, or like the common ancestor of what, like what came first. And again, these are all grounded on sometimes presuppositions, pre-assumptions that we have. And the scientists, many times they observe the data, but they have already they believe, they have their own convictions, they have their own beliefs, and they believe that this is the case, right? That this actually is true. And therefore, when they see the fossil records, they analyze them, they interpret the data based on what they already believe happened. And therefore, that's a bias that sometimes leads to an interpretation that might not be the correct one. Again, this happens to everybody, right? Anyone is prone to bias and has convictions and beliefs, and that's why we cannot stay completely objective, not even scientists. And so we always have to remember about this. So as we use words and how we define things. But again, it's interesting that this particular article is actually contradictory, because here Galapagos finches are used as macroevolution example, but actually before they themselves use it as a microevolution example. So yeah. And yeah, that's what I wanted to show. And now again here, as I said before, I'm gonna go through these different examples, right, that many times is used to support this is example of macroevolution, right? We already seen the Drosophila. Let's take a look at these other ones. HIV resistance. So a specific 32 base pair deletion in human CCR5 confers HIV resistance to homozygotes and delays its onset in heterozygotes. That's the difference I was saying before, right? Homozygotes, you have this deletion in both alleles, right? Heterozygotes instead only have one allele with this deletion, the other one is working properly. And again, here there are different explanations, right, that evolved from the bubonic plague or sometimes the smallpox, right? There is another one theory that says that it's from the smallpox. Doesn't really matter. The idea is that when this particular gene here is deleted, so it's not present, should be present, but it's not there, well, this gives resistance to HIV, so to the human immunodeficiency virus, and to the homozygotes. On in heterozygotes, it gives delayed onset. And now, why? Have you ever wondered why do human beings with this mutation, right, with this deletion, with this part of DNA that is not present in this particular gene, why is it that they are immune or are resistant to HIV? Well, because this particular protein is actually what the virus uses to enter into the cell and to reproduce, and therefore to affect the organism, right? To then create the disease in a sense. Remember, the HIV virus many times you can be HIV positive for a very long time before you start manifesting the symptoms of the AIDS, which is the actual disease caused by the HIV virus. So already, but what happens is that because this particular protein, right, this particular gene that codes for this protein is deleted, the virus cannot do that. It cannot enter into the cell. It cannot duplicate itself. And therefore, the organism is immune to it. That's it. It's very simple. And that's why heterozygotes, where they do have this protein, but it's less produced because of the fact that it's only half of it, right? Half doesn't work, half works. In this case, it's delayed. So it's like it takes more time for the virus to enter or to replicate itself and to do its own cycle. So the virus cycle replication is halted because of this protein being deleted, right, or not working properly, and that is what confers resistance. So again, I just want to show you this because this is not a gain of function, even though it's something that gives an advantage, an evolution advantage. But this is not a gain of function. This is not a new gene. This is not a new like structure that is being created. It's not like the human develops a resistance to the virus in terms of being able to repel it or being able to, I don't know, change the structure of a protein, still having it. No, it's just a protein that should be working a certain way that does not work or is not present because of this mutation. And well, it happens that that particular protein is what is used also by the virus. So because it's there, you're immune to it or you're resistant to it. And again, this happens the same for the malaria. Again, because the harmful mutation is the sickle cell disease, right? A blood disorder in which the body produces an abnormal type of oxygen-carrying substance, hemoglobin, in the red blood cells. So they are in a different shape. And now it happens that 1/3 of all the inhabitants of sub-Saharan Africa carry the allele, so the part of the gene that is mutated, right? Because in areas where malaria is common, there is a survival value in carrying only a single cell allele. So the ones that have both alleles, they ended up dying because of the disease. While those who are heterozygotes, so those who are carrying only one single cell allele, so they have the disease, but it's not that strong. They have a survival value given. Why? Because those with only one of the two alleles of the sickle cell disease are more resistant to malaria, since the infestation of the malaria plasmodium is halted by the sickling of the cells that it infests. So again, because there is this problem, the red blood cells are not as they should be, but they are sickling, right? They are like half. Then the plasmodium cannot use them to replicate itself for the actual malaria, the disease, to spread. And that's why these people are resistant to malaria. So again, you can see that it's not actually a new function, isn't it? It's not a new way in which we can repel the plasmodium or we're immune to it. It's just that the particular thing that the plasmodium uses for its living cycle is not working, is mutated. There is an issue. And so you're immune to it. But again, remember that you still have the problems of the disease. Maybe because you have only one single allele, you don't have that many problems, but you still have something that is not working as it should. But in this particular case, it's giving you an advantage. But again, I want you to show you these all examples. This is microevolution, right? These are not superhumans or another species of humans that have acquired a new function to resist to. No, really. It's just something that's not working properly, and it happens to be the way in which also the virus or the plasmodium in this case uses to replicate itself, and so you are resistant to it. For bacteria also it happens the same. So practically all bacteria develop antibiotic resistance when exposed to antibiotics, and again they have already some mutations that get selected under antibiotic selection. So bacteria have already mutations that made them more or less prone to resist to certain antibiotics, and when you use antibiotic, the bacteria that are resistant to it are the ones that are going to survive. So they're going to reproduce, and so then you have all those bacteria that all are immune. And again, this is a huge problem in hospitals because you have these super bacteria that are immune to tons of different antibiotics, and we cannot kill them. So it's becoming serious. That's why antibiotics should only be used when there is actual infection and not be used just whatever, right? Because otherwise you risk of developing resistance to the bacteria to become resistant. And now again, why are the bacteria resistant to antibiotics? What happens? Again, the protein that the drug uses to kill the bacteria is mutated, is changes in shape, changes a little bit in function, and so it cannot be used anymore by the drug, and so the bacteria is immune. That's pretty much how it happens. And then there is last one here on lactate persistence. Again, the mutation allowed humans to express the lactase enzyme even after they are naturally weaned from breast milk, allowing them, the adults, to digest lactose. And again, this is something that there is a regulatory system in this case. So this enzyme here originally was produced only in the first few years of age, and then it was regulated negatively, so it was not produced anymore. But then a mutation and the regulatory part of DNA then happened that then made that this enzyme is still created in after the first years. And so even adults can digest lactose, even if they shouldn't in theory. So again, another example of not really a gain of function, not really new function or new genetic material, just something that it's already there, that is mutated, that changes, and because it changes the regulation of something else, well, that particular thing that is not regulated anymore can actually be used now, and it's of some advantage. So yeah, that's the idea. So you can see how we can explain all these examples. Many times also it's given the examples of the bacteria, the Enteric coli. I don't know if I have it right here. That can actually become, like, they become larger. They become, I don't know where I have it. Yeah, the salmonella, maybe this one. It might be this one. But like they start like developing more. They start being a bit larger, and they start like being able to use also an amino acid as food source that before they couldn't. And so scientists say, yeah, this proof macroevolution. The bacteria are becoming larger. They are able to do all this stuff. And then you actually take a look at why they are able to do that. They became larger because the protein that was regulating the size was mutated, so it wasn't regulating it anymore. So that's why they became larger. And again, being larger does not mean an evolutionary advantage, because sometimes maybe being smaller is better. Or again, the fact that the bacteria reproduce too much is not always a good thing, right? Cancer cells also reproduce too much. So the genes that are regulating the expression, so how much a cell reproduces, they go bad. And that's why the cells are reproducing too much, and that's why cancers arise. But again, just because something is reproducing lots of times doesn't mean that it's actually good. So again, see how it depends. And again, these bacteria being able to use another food source, or like being able to code for a certain metabolic rate, again, because of certain proteins that were functioning mutations, they were alterated. And so in their alteration, they were able to bond even to things that before they couldn't. But again, here, it's not like a new function, right? Or new structure that happens. It's just a protein that before it was fully functional, starts working bad. And because it works bad, sometimes this bad working allows the protein to maybe bound or to allow a different pathway. And that it might be beneficial. But again, remember, it's always mutated. So it's not doing its original work, but it's doing something different. And again, I would argue that how can we know which is beneficial and which is not, right? So to how we're quantifying mutations in order to know, or DNA changes in order to know what is good and what is not good, what is harmful, what is beneficial. And again, this is very subjective, right? Because we might be seeing a larger bacteria, oh, that's good. I don't know. How can you say that's good, right? Bigger does not mean always better. Or the ability to maybe not be to have a different pathway, right? Yeah, maybe the protein was actually coding for something else. So yes, it's true that you might be able to have a different pathway now, metabolic pathway, but maybe you cannot do anymore something else. So in another condition, it might be something detrimentary. So again, it's very complex, but this just to show you that not always this the things that we observe or the things are used as evidence of new genes evolving is actually evidence of macroevolution. Yes, microevolution, yeah, but then macro, these big changes that then create more complex new structures. Evidence for this? There is none. And again, prove me wrong. Do your own research and see if there is actual something that evidence that suggests that this is true, that this actually happened, that there is evidence of this. And again, there is a lot more that could be said. I'm not going to go into that much detail. I'm going to just show that this new gene origination that we have is actually always from selecting parts of DNA that is already there. So even if it's non-coding, right? But we do not know if that non-coding part of DNA maybe was important for something else. And again, we might not know also that when we are evaluating, yeah, that's a new gene because we are comparing it to something else that we already know. And again, but again, it might not be that it's always beneficial or that it's always good to have it. And actually, if you take a look at yeah, this one right here, having this microRNA, right, that's been coded that before it was not coded, is not always good, because I think there is this article here. Yeah, this is someone done on Escherichia coli. This 25% enriched, right? I remember I read this article. So so it's interesting to know, like, just because some of them can reproduce more quickly, right? And something like that, like, it doesn't mean that always is a beneficiary, right? And actually, if you look at this, these mutations, right, that occur in the non-coding regions of the DNA, that they can alter the abundance of messenger RNA, and potentially facilitating cell proliferation, is not always the case that they're good. Actually, they're actually linked to certain types of cancer. So you can see how not always, like, being able to have more cell proliferation or increased metabolic rate is always good. So it depends. And again, that's why I wanted to show you that just because something mutates in an area that we might not consider like something that was useless, right? Or what it says here, let me see here. Yeah, because the non-coding regions, right, for a long time we believed that they were just not having any relevance. They were just there just to protect from mutations, but actually they are very important because they regulate the expression of the coding parts of DNA. So mutations there, if happen in certain scenarios, could actually lead to problems in regulation of that particular gene. And again, that could lead to cancers, that could lead to proliferation, that could lead to issues that we have studied, that we know. So again, everything from the DNA is important. So again, I hope that I was able to show you how rich and how fascinating this is. And again, the issues. Many examples are given for macroevolution are actually examples of microevolution. And again, that there is no actual evidence that suggests this took place. Even in the fossil record, even the transitional fossils that sometimes are used to affirm that, yeah, see, this is a species that has similar features to this one and also to another one, right? It's kind of in the middle. But how do you know that that's actually a complete independent species that just had those particular features, right? How can you know? We don't, because it's just fossils. It's something that we recreate. Many times we do not know how these fossils were, their behavior. We just have the bones. So we cannot really make that many considerations about them other than maybe their shape, their morphology. But again, sharing similarities doesn't always mean like correlation or that there is a common ancestry between them. And again, so these are just some of the issues. Again, I'm going to put all these articles in the description below. I really hope that you can read them and you can look for yourself that many of these examples, example, this one here for the tryptophan acid, it's interesting to see how there was the input of the researchers, of course, right? So there is here is not completely random. It's kind of a guided in the lab. But anyway, the gene, which has this weak function to produce tryptophan, was enhanced. And so at the end of the year, was like 300 generations later, the bacteria had one gene that did the original job, and a second that had evolved a new primary function, manufacturing tryptophan. And this was because of this fact that there was already a gene that had this primary function and that had a secondary, less important function. And again, just by overworking this secondary function, they were able to reach to this idea that it became a primary function. Again, it was still manufacturing tryptophan. So that was the gene that was missing, right? So one strain that was missing the key gene for the expressing the amino acid tryptophan, right? So this was missing, but there was another one who had a weak ability, right, to produce tryptophan. And it's important. It's interesting that the function was still manufacturing tryptophan. So why didn't the bacteria develop the ability to manufacture, I don't know, valine or leucine or another amino acid? Why it was still tryptophan? Because again, it was already there. It was already in the DNA to be able to have that code. So the environment in which the the researchers put these bacteria, forced him in a way to maybe express more of that gene to be able to adapt to the environment, to be able to then start producing more tryptophan. So again, you can see how there is no ex novo or new genes being created. It's always the material is already there. It's just acted on by the environment or by natural selection or by the mutations. And again, there is an alteration of something that's already there, but there is no evidence whatsoever that these new genes, right, can have complete new functions that weren't even there proposed. So again, there is the emergence of these genes that have been seen, right? But then can they actually lead to something that's actually complete new function that wasn't even there? You know, because again, it's mutations happening in the non-coding parts, mostly non-coding parts of the DNA. That again, that we see, that we can compare because we know that particular genome sequence. And again, remember, because the DNA is a sequence, right, of this, it might be that another part of DNA is very similar to this, just with one letter or a couple of letters difference. So if a mutation happens there, then it could be that it becomes the same, right? So it becomes the same like as the coding part. Again, I hope that you can really read these articles for yourself. And you can do your own research and let me know what you think in the comments. So I hope you enjoyed. Thanks for watching and see ya. Bye bye.